So yeah, pleasure to be here. Also, thank you to Jürgen organizing to invite me to give this presentation. For disclaimer and full transparency, I am co-founder of Tesorai, which is one of the sponsors of this event. So hopefully that didn't influence that decision. And I hope I was invited here based on merit and interest in what we are working on. Maybe just for some background or first agenda, I'm going to cover a few topics all related to Tesorai Search. I'm going to spend quite a bit of time on giving you background on challenges, how we think about challenges in computational mass spectrometry. So if something's going to be unclear along the way, please speak up, ask questions. I would really like you to understand what we are doing, why we are doing it. So yeah, don't be shy, just shout out. For some quick intro on Tesorai. So basically, spending some time at UCSF in computational biology and then in big tech for several years, that gave me quite unique perspective in terms of how to think about software, how to think about modern AI, ML tools and software for analyzing big data. And working in that space for a while, I've noticed few gaps. The way I think about data workflows is that you start with raw data as collected by the instruments. Then the first step that needs to happen is to pre-process that data and turn it into structured tables, for example, protein expression tables that you're familiar with. And then the next step is to analyze those tables in order to generate new insights, find new biology, find novel biomarkers, drug targets, or develop drugs themselves. And yeah, just working with people, collaborators, friends, colleagues in this area, I've noticed that most of the efforts is usually spent on the latter part, on the second stage, analysis of the data. The first step, pre-processing, is often overlooked, both in terms of how many people do we have working on that, how much resources are being funded and put into these efforts, and that really shows in the results. So when you look at these data workflows, there is tremendous amount of data being lost during the sample or raw data pre-processing. We believe, or in many cases, some of that data is trash, it's noise, it's garbage, and it should be discarded. But we believe there is also lots of biologically relevant information in what's been lost and discarded in the pre-processing. So with Tesorai, this is exactly what we focus on. We are building AI, ML, and software solutions to generate the most impactful insights from every byte of data that many of you are working super hard to generate along the way. And if you're not working hard, you should, because that's important. So yeah, today I just want to give you a story on what we are doing in mass spectrometry space. The goal for Tesorai is to be much broader than that, to go into multiple omics platforms, but we are starting with proteomics mass spectrometry. The work wouldn't be possible without great colleagues, collaborators, Juan, Shamil, Jürgen, who are here. Maybe also some of you know Shivani, who used to be in Jürgen's lab, and collaborators at NYU as well. So going back to how much data do we lose in data pre-processing stage, mass spectrometry is a poster child of that. For example, we still have, in some cases, up to 75% of spectra that remain unidentified, which basically means that we only see, in some cases, again, up to 20–30% of analytes that are actually present in your data. The rest just isn't available to us. And even for those that we can identify, quantification can be off, in some cases by orders of magnitudes, especially for low abundance proteins, which arguably are the most biologically interesting. So there is lots of work to be done. Thinking about why that's the case, some of it goes back to software that we are using being primarily written to run on workstations, in many cases, Windows workstations. The software is optimized for speed. It needs to process massive amounts of data in practical amount of time, and not necessarily accuracy. And what was really interesting to me is when we are talking to labs, few of those, or quite a few of those, actually pointed out to us that data pre-processing has become a bottleneck. They started to generate data a lot more quickly than they can process it, which in this day and age with cloud compute, with AI, ML, and so on, just shouldn't be the case. So that's what we are trying to fix. And before going into that, I just want to give you, say, quick background in terms of how we think about software for interpretation of mass spectrometry proteomic signal. So we have heard in this summer school quite a bit about database search. That's one way to analyze the data, but there are also a couple others like spectral library search and de novo sequencing. but the database search is still the most widely used. It's also going to be the most effective for some time, both in terms of time that it takes to process data and accuracy. Database search has gone through a couple generations of evolution. The first generation, or Gen 1 as we call it, are algorithms that have been around for quite some time. Examples of that would be Andromeda Score, Sequest, Comet, and so on. What's common to them is that they use this simple barcode matching. So basically the way they work, can you see the cursor? Yeah. So the way they work is you have a real spectrum on the left side, you have a database of protein sequences on the right side. The software is first gonna in silico digest those proteins into peptides, generate theoretical spectrum spectra or spectral libraries. And in this particular case, they are nothing else but a list of MZ values without intensities for the most common Y and B fragment ions. So no non-canonical fragmentation is going into that. And then there's going to be a relatively simple equation measuring correlation between theoretical spectrum and the measured spectrum. And the good thing about this software is that it's fast. You can process data locally, but by ignoring intensities of Y, B fragment ions, by ignoring non-canonical fragment ions, that leads to inaccuracy or data loss along the way. These scores usually also aren't very well calibrated. So for example, if you take one spectrum and peptide and get a score 50 for it, and another spectrum and another peptide and also get a score 50, those two scores are not necessarily apples to apples comparison. So the scores are not necessarily well calibrated. Then a few years ago, a lot of it driven by the publications like ProSet or DeepMass that we have worked on, Gen 2 algorithms came along with the introduction of AI/ML. They've added a lot more complexity to address some of the challenges that I've mentioned. So they still work by having a measured spectrum on the left side and protein database on the right side. But now, let's say the theoretical spectrum generation is replaced with, or adds one more component to it. So here they are using AI/ML tools to predict intensities of those Y and B fragment ions, which improves on quality and accuracy of spectral libraries. And then they do the following. They manually design features that describe similarity between theoretical spectrum and measured spectrum. And these features are handcrafted. These features are not necessarily just related to the spectrum comparison, but also take into account peptide properties like peptide length being one of them. And then they feed all of these features into tools like Percolator. And Percolator is another machine learning model that's trained on the fly on your specific data set. What it does, so basically, if we go back to this table of manually generated features, so you have let's say, a table with rows that are PSMs, some of them are with target peptide sequences, and then you have columns as features. So what this, let's say, machine learning model here is going to try to do is to find a way to separate targets from decoys. And again, it does that on your specific data set, so it's trained on the fly. And then you'll use that same data set to estimate FDR and do analysis downstream. And as a data scientist, that's a pretty poor practice because it can lead to overfitting. Yeah, so just to give you, a few examples for why that is the case. But maybe before going into that, I was really glad that some of the people started to bring up this issue of overfitting. This is from a recent publication from Bill Noble's lab. He also spoke about that at ASMS. And I was quite surprised to see multiple posters at ASMS talking about this issue with Gen 2 algorithms in particular. So what you see here on this left side is a result of entrapment analysis. Entrapment analysis basically, I'll try to be short, but basically estimates the true FDR in your outputs. So the problem with Gen 2 algorithms or overfitting in general is that when you think you have, let's say, 1% FDR in your outputs from the software, you in fact have a lot more than that. In some cases, up to 10%, which is what we are seeing with some of the Gen 2 algorithms. So having so much inaccuracies in the output results can lead to bad science downstream, and something that definitely needs to be fixed. The reasons or the sources of this overfitting, well, there are a few of them. One is incorrect training and validation data splits. So when you do machine learning, when you train a model, you want train set to be independent from your validation set. You don't want to have examples that are shared between the two because that can bias not bias the model necessarily, but bias how you estimate how good your models are. And that's actually happening with Percolator. That's something that they're trying to fix. But the problem here is that for a given precursor, you can measure multiple scans, for example, at different retention time points. And when you split the data randomly into training and validation set, you can end up with having some scans from the same precursor in train and others in validation. And that can, again, bias in terms of how you're going to evaluate the model that you are working on. So that's one source. Second source is decoy-specific information leakage. So an example of that would be how we generate decoys. For example, if you look into Diane's specification for how they generate decoys, they turn the second amino acid and second to last amino acid into another amino acid. And that mapping is usually going from any amino acid to fairly small amino acid like leucine, alanine, and so on. And if some of the features that you are feeding into Percolator depend on properties like retention time prediction or ion mobility prediction, you can imagine that changing any amino acid or changing especially large amino acids for a smaller amino acid is going to create shifts in distributions between targets and decoys. So your model is going to learn how to separate targets and decoys, but not based on what truly matters, what you really want. It's just going to it's going to start separating targets from decoys based on these undesired properties, so some sort of leakage that's happening in the data. And then finally, leakage can also happen, or overfeeding can also happen when you have a small data set. So these models like Percolator are not super large, but they are still complex enough to overfit on smaller data sets. So you only have few runs or small, let's say, raw files. I would be cautious by using them because, again, they can overfit fairly quickly. So yeah, so with all of these challenges, we wanted to rethink how we do search and build something with best practices using modern AI ML. Sorry for throwing that buzzwords. I'm becoming allergic to it, but that's what we are doing. And compute practices in mind as well. But instead of being closer to Gen 2, which, as you have seen, is a very complex set of steps that need to follow with risk of overfitting, we want to build something that's going to be closer in spirit to Gen 1 algorithms. So basically, we still want to have spectrum on the left side, database on the other side. Here, we can add hits from de novo search as well, and then just have one model that's going to perform both theoretical spectrum generation and correlation metric development. And here, in this model, we are not going to explicitly tell it to generate theoretical spectra. We are going to train it in a way where it can do that implicitly, or it may not do. Like the model is going to figure out how to put the best peptides together with a given scan. And we need to do quite a bit of innovation here to make that happen. So again, the goal here is to build a single model. We don't want the space to be even more fragmented by having one model for different acquisition approaches for different enzyme digestion and so on. We just want all of these to be covered by one model. So TDA, TMT, DIA, having different PTMs, instrument manufacturers, enzyme digest, or sample prep protocols. We want all to be supported by one model and one model alone. We want that model to be end-to-end model. So we don't want it to go through multiple stages, as is the case for Gen 2 today. We just want it to go end-to-end. So peptide and spectrum in and a score going out. The next bullet point is very critical. We want that model to be entirely pre-trained. So we no longer want to support behavior where we train a model on the fly, because if somebody is not familiar with overfeeding, they are going to be they're going to be at risk of running into that. So we want this model to be ready out of the box. When you bring your data in, we're just going to use the model. We're not going to do any training or fine-tuning on it whatsoever. And a bonus here would be that if we can train this model without using decoys, then we would have very small risk of overfeeding. The problem with training model on the decoy, especially when that model is large, is that the model can start remembering what's a target sequence and what's a decoy sequence just from the sequence alone. I've been tracked by this large model many times, so that can happen. We also want the model to use entire spectrum as an input, not only the canonical fragment ions, but everything that's been measured. And we want to build it in a cloud environment. not only because this model do require more compute to be executed in a reasonable amount of time, but also because I think we can speed up the processing of mass spec data considerably by doing so. So yeah, so that's what we have set out to do. Again, just to repeat, we want to build a large pre-trained end-to-end model that takes a spectrum as an input, peptide as an input, and produces probability of those two being a good match. And because some peptides are going to be decoys, some are going to be targets, we can do standard FDR estimation, just using target decoy competition. And we can also follow on with post-processing to do quantification and protein inference in a standard way. To build this model, we have curated and reprocessed large amount of data, so several tens of publicly available projects, and labeled them with MaxQuant, reprocessed them with MaxQuant to get the labels for what peptides should go together with what spectrum. And currently, the slide says 100 million, but we have over 300 million training examples, where each training example is a PSN, basically. The model is fairly large. It's probably by far the largest thing that you will see in mass spectrometry today. It has over 10 million parameters. And it's trained in a unique way where it's not using any decoys during the training. So we are developing, let's say, a loss function that's more based on ranking and semi-supervised learning, and it doesn't depend on decoys. So the risk of running into overfitting, again, is minimized to as small as possible. Cool. So we have our first model built, and I'm just going to show you some of the results. So the first thing that we want to make sure is we want to make sure that this model is going to be accurate, that it's going to accurately estimate FDR. And just looking at the same curves, following the same procedures as proposed by Bill Noble in that paper, we are seeing that our model is spot on. So when we say that there is 1% of false positives in your output, that's exactly what happens. So the model is accurate. The model also seemed to be comprehensive. When we applied to left out data set from proteome tools, we see that we can increase coverage, or how many peptides, synthetic peptides, do we identify in that data set quite a bit. So compared to current approaches that detect 75% of those synthesized peptides, we can increase that number to 92. Or let's say, if you're talking to venture capitalists, you say that you've reduced the missed identifications by 3-fold. So very significant result. But what was really exciting to see is that we can increase peptide identification without using percolator. So just using pre-trained model off the box. What you're looking at here are the results by running our model on 4 different benchmark data sets. So each individual block here is different data set. One note about these benchmark data sets is that we haven't cherry-picked the data. We have just used the data sets that others have reported. For example, ProSeed paper or MS Booster publication. We took those same data sets just to make sure that we are not finding some data sets accidentally where we would do much better than others. And what you're looking at, these graphs are peptide identification counts, where the leftmost is for MaxQuant, the next one would be for ProSeed plus percolator, MS Fragger plus percolator, and then our Tesorai Search. The blue colors in these lines mean or denote the number of peptides that are shared with those identified with MaxQuant. Green ones are new additions, and the red ones are losses compared to MaxQuant. So all in all, what you see here is that Tesorai Search boosts identifications anywhere from 5% on, let's say, standard HeLa triptych digested sample, all the way to 90% compared to MS Fragger and ProSeed for some of the immunopeptidomics data sets. So we also wanted to make sure that our model generalizes well, as mentioned, by applying it to what we call out-of-sample use cases. So our model was entirely trained on thermo data, and we wanted to make sure that it works on data from Bruker timsTOF. And what you see here is the same or similar boost in identifications also on timsTOF data that our model did not see during the training whatsoever. Similarly, the data that we have, the data set that we have right now has very small number of TMT labeled data examples. And as you can see, it performs really well on that type of use cases as well. And for anyone that's interested in single cell proteomics, we show that our model performs really well on low input data sets as well. For example, from 1 cell all the way to 50 cells, we see significant boost compared to other approaches as well. So there's quite a few questions that we are getting. Are those new identifications real? And we have few ways to answer that. One, we are always doing standard target decoy competition. So we're controlling for false positives that way. But one thing that we also decided to do is to work with experts, human experts that are reading these mass spectra, and give them few examples from, let's say, cases that multiple models agree upon and cases that are uniquely identified by our Tesorai search. And we've asked these human experts to basically classify or categorize those PSMs into three categories. One would be this PSM is incorrect. Second one would be this PSM is correct. And then there is one in the middle that they called passable. They call something passable whenever they are looking at chimeric spectra, so spectrum that's generated by multiple peptides being co-eluted, so they can't explain, or let's say the sequence of a given peptide does not explain all of the peaks in a spectrum, but they are still happy with what they are seeing, and it's likely a correct annotation. And what was really cool to see from this study, and this work is still in progress, so we continue to collect more data on it, but let's say as some sort of interim readout, what was really exciting to see is that so far they haven't labeled one single case as incorrect. So it seems like our model is doing the right thing, correctly putting spectra with the right peptides. So going back to where these identifications are coming from. So what was really interesting to see is that many of the hits that we are proposing are also top scoring hits by Andromeda. The problem is that for Andromeda score, it didn't make it through 1% FDR. So in some cases, that PSM was just below 1% FDR, between 1 and 5%. But there are also a few cases where, let's say, the model, so this is the pink distribution here, where Andromeda score thought that they were below 5% FDR. So this goes back to when I said that current scores, Gen 1 scores, are not very well calibrated. And it seems like more complex models is being able to deal or calibrate these scores in a better way. This is also what Percolator does in Gen 2 approaches, but it does it in a way, again, that's prone to overfitting. So I think a lot of what is happening here is just calibrating the score properly. And yeah, also one interesting observation, new identifications most of the time have this shift towards lower abundances of precursors. Meaning that, let's say, with Tesorai Search, you're going to be able to identify even lower intensity signal than you would have with standard approaches. Yeah, and then to bring it back the full circle, we wanted to see whether having a better search algorithm is going to be able to uncover new biology, basically. So to this end, we looked into a data set that was generated by Nevan Krogan lab at UCSF a few years ago. It's a data set based on a bunch of pull-down assays between breast cancer oncogenes and human cellular proteins that might interact with those oncogenes. It's a data set of about 700 files, so pretty massive data set. So when we reanalyze that data and look at what we've been identifying, what was really interesting to see that we identified 100% more proteins than what they have reported in the paper. And going back to validating some of these results, what was a nice validation, let's say, is that by increasing how many proteins we see, we also increase the overlap with what's known about those interactions as well. So just another way of validating that these results might be real. And that's on it. That's it on the results. Maybe just a few words if we have time. We do have time. Okay, on the platform itself. So I talked quite a bit about two things that were important to us: accuracy, so making sure that we are not overestimating or underestimating FDR. Comprehensiveness, so we would like to see as many peptides as others or more if possible. But there is also a third category that's important to us. We would like to package this model into something that's going to be as user-friendly as possible. Yeah, so we've decided to build Tesorai Search into a web platform. Search, currently, it's a cloud-based platform that hopefully it's going to be fairly easy to learn and use. So basically the way it works, and I will give you a link later where you can try it yourself. But basically the way it works is you can just drag and drop raw files into a web browser. Because we are having one large model, there is fewer parameters to fine tune. So there are really just a few parameters that you can fine tune. For example, pick an enzyme, digestion enzyme, and so on, and then start the job. And in some time, you're going to receive an email that the job has been completed. Hopefully, it's not going to be a failure. And then you can download the results. The results currently are fairly minimalistic, but we would welcome any feedback in terms of how we can improve there. Speaking of time that it takes to process the data, yeah, the platform is extremely scalable. So we ran it on hundreds of files. Here we are showing an example with 500 files that were all processed in less than 1 hour. And this is from, let's say, that Nevan Krogan's database data set that I've mentioned. And obviously this number 1 hour or so is going to depend on the size of files, the size of FASTA file that you're using for the search, what enzyme have you chosen. So it's not always going to be under 1 hour. It might take a couple hours or a few hours with immunopeptidomic searches. But the point here is that the platform is scalable. So it's going to roughly take the same amount of time whether you upload one file or thousands of them. Doesn't matter. We are a very young startup, but moving quickly. We've been adding features on a monthly basis. So we've launched the company in September last year, just went through our first anniversary. Our first model prototype was ready in January. The whole platform in beta form was ready for ASMS in June. And then just a month ago, we've added TMT support, protein inference, and quantification. We're going to be adding some exciting stuff in the future as well, like PTM and DAA support. And perhaps most excitingly, integration with MaxQuant. But what I also want to say here is that we are building these for you. We would like it to be easy for you to use, useful to you. And would appreciate any feedback that you have. So I can assure you that it's not going to be perfect as you give it a try at the beginning, but we would like to build it that way. So any feedback that you're going to have, please reach out and we'll try to take that into account. Speaking of feedback, I don't know if you're aware of this blog, News in Proteomics Research by Ben Orsborn. I've been told that this is the proteomics blog out there. It's really useful to read because it covers lots of, Ben covers lots of developments, new developments and stories. And yeah, he also picked up our preprint last week and gave us pretty solid reviews. So seems like we are legit to him. He had no problems learning about using the platform. And he wasn't sure, but he thinks that the number of proteins that he saw was the largest than any combination of tools that he has used on this data set that he's familiar with. He also said some constructive feedback, how we can improve in terms of fast uploads and stuff like that. So I don't want to encourage to only share positive feedback. I would really like for you to share constructive or negative feedback as well in terms of how we can improve. So yeah, so this is a QR code that's going to take you to, I think, one of the two URLs below. If you would be so kind to give it a try, please do so. Any issues that you're going to have, any feedback, any comments, any questions, feel free to reach out to me directly and would love to chat about your use case and how to optimize the results that you're working on. With that, I always like to finish talks with one more thing. So we are super excited about what we have built with Tesorai Search. Again, it's early days, so lots of improvements to come, but we're also working on some pretty cool stuff, I think. We call them instant search and incognito search. The reason we can build this is because with the way we have built Tesorai Search allows us to do a couple of really cool things. The first one, instant search, is basically us imagining a world where you can do search instantly. So when you upload the data, you just select what you would like to search for and you get the results instantaneously, basically within seconds. And if you want to add phosphorylation as a PTM, you just click another button, rerun it, and get results back instantly again. So that's where we would like to go with instant search. Incognito search, on the other hand, is we are building that for a specific use case. This would be built for, let's say, 2 or more arm studies. So for example, if you have a group of samples that are control and another group of samples that are treated in some way, and you're looking for differentially expressed proteins in one group or the other, this tool will allow you to do that, but in a slightly different way. So if you do that today, basically you're going to be limited by identification tools. So if you can only identify 20 or 30% of the spectra, you're only going to be doing differential analysis on small fraction of proteins that are actually present in your sample. And as you can imagine, that's going to limit the success that you're going to have with these sort of studies. What we are building towards with incognito search, though, is a way to utilize all the spectra, all the information, so 100% of the data that you have in there. And just to show you what might be possible with this approach, and again, this is very early prototype that we have here, so it's more of a demonstration of what is going to be possible with these sort of approaches. But if we look at an immunopeptidomics data set, this immunopeptidomics data set is based on tissue from breast cancer tissue and adjacent healthy tissue. And it was published a couple years ago. Data was analyzed by state-of-the-art platform. It was MS Fragger with open search with some machine learning on top of it called Promise. So this data set, they were able to identify 53 hits. If we use incognito search, we are able to increase that by 6-folds, which is, I think, very drastic. And the reason this is important is because when we think about biomarkers or protein targets, the numbers matter. So going from this early stage discovery through translational science all the way into clinics, many of these biomarker candidates are going to drop out for one reason or another. So if you start with 50, your chances of success are going to be something. If you start with 300, the chances of at least one making it all the way through the pipeline go up considerably. So that's what we are trying to build towards. And hopefully it's going to work. With that, thank you all.